Welcome to my YouTube channel Fast Track Learning. We are going to learn about chaperone proteins. What are chaperone proteins? These are the molecules in cells that assist in folding and assembly of other proteins. They are itself protein and they help in the folding of other proteins. Now the proteins have 3D structure, so it can perform its function properly. I have also shown a type of chaperone protein, which is a chaperonin protein. It has a beta barrel shape. It has a cap. When a protein enter in it, it would be converted into a properly folded protein. Now we can see the functions of chaperone proteins. They prevent misfolding and aggregation. They prevent aggregation because they help in the correct folding of other proteins. They are itself proteins, they help in the correct folding of other proteins. In this way, they prevent misfolding and aggregation of proteins. And also, if the protein is already misfolded, so it help to refold that misfolded protein. In this way, it prevent the misfolding of proteins. We can also see here the chaperonin, which can be taken as an example. This because this is a type of chaperone protein. Now let's see the all types of chaperones. These are chaperonins. These are HSP70. HSP stands for heat shock protein. HSP90, heat shock protein 90, small heat shock proteins. First of all, we are going to learn about chaperonins. Chaperonins are the specific class of chaperones that provide a secluded environment for proteins to fold correctly. They have a size of 800 to 900 kilodalton. They requires ATP to perform their function. They form a large barrel-shaped complex to encapsulate folding proteins, um, ensuring an isolated environment for correct folding. This method is specifically suited to large complex proteins that need extra space for proper folding. So they provide an isolated environment. They provide a perfect environment for folding of the large proteins, we can say. So now we are going to see the example of chaperonin, which is GROEL, which is HSP60. It is a chaperonin. It is found in bacteria. Its function is the proper folding of protein. Now let's see heat shock proteins. First, we are going to see heat shock protein 90. It is heat shock protein 90. Its weight is 90 kilodalton approximately. It requires ATP to perform its function. It keep the cell structure stable. It means it keep the cytoskeleton stable. HSP90 help the steroid hormone receptors to work correctly, which help the cell to respond to hormones like estrogen, testosterone. It is always present in the cell. It is not only stimulated due to stress or shock. It is always present in the cell. It helps the correct folding of proteins. And now we can see its example. The example of HSP90 is GRP94. It is localized in ER. It helps in the correct folding of the proteins. Now we are going to see another heat shock protein, which is heat shock protein 70. Heat shock protein 70 and its weight is 70 kilodalton. It requires ATP. It assists in folding. It helps the proteins to fold correctly. It is a molecular chaperone because um it protects the proteins. It helps in their proper folding. Um, it repairs the proteins which are damaged. If the damage is irreparable, it helps in the breakdown of the damaged proteins. And it is always present in the cell. It means it helps in both conditions, normal and stressful condition. So it is very important. We can see its example. The example of HSP70 is DNAK, which is found in bacteria E. coli. Now we can see small heat shock proteins. They have less molecular weight. They are approximately or we can say they are in a range of 12 to 43 kilodalton. They don't require ATP, making them energy efficient during stress. They bind with partially unfolded proteins. They help in the proper folding. but they are not always present. They are only active in stressful conditions. Um, we can see the example of small heat shock proteins, which is HSP27, which is found in cytosol. It helps in the proper folding. Now we are going to see the mechanism of heat shock proteins. We have um, the protein which can be nascent, which can be unfolded, a protein which is misfolded or denatured. So when a chaperone bind, if a protein is unfolded, it is nascent, it help in the proper folding of protein. But if a protein is misfolded, it prevent the aggregation and help in the proper folding of the misfolded protein. If a protein is denatured, it help in the refolding of denatured proteins. So these are the functions that are performed by the chaperone. We have previously discussed different types of chaperones that help in performing different functions by the chaperone proteins. Thank you. I hope you understand. If you have any query, you can ask. Please like and subscribe the channel as well.